Each year, Microsoft Research hosts hundreds of influential speakers from around the world, including leading scientists, renowned experts in technology, book authors, and leading academics, and makes videos of these lectures freely available.
So it's a big title, but I'll try to fill it with some stuff. So I'm at the University of Warwick and running a wet and dry lab. The wet part is very new, so don't ask me questions about that. So what we're interested is basically this very simple statement that biology is a result of evolution. So we like to understand how this process of evolution works and what kinds of things it can generate and what kind of processes drives it. And obviously we know about selection, so fit gets fitter, but obviously there are additional processes in place, and even if it's just driven by selection, then we're interested in understanding what those selective pressures are. So you can abstract the evolution by this concept of a genotype-phenotype mapping, where genotype represents all the parameters you can think about that can change a particular trait, and your trait is this phenotype. So you can have this multidimensional mapping between these things that you can tune, or the evolution can tune, and the resulting higher level behavior that comes from that. So you can put different species on this kind of a map. Obviously, this is not multidimensional enough, but obviously people will be there and all the other organisms. So we tend to work a lot on sort of microbial systems because they're easier to work with, but we also aspire maybe at some point to understand things like humans. And there are interesting things happening. You can, for example, imagine a particular region of that genotype space could correspond to very similar phenotypes, although they will be different. So, for example, the genotypic pool of Italy have produced great politicians, but it also produced great scientists. So I also like to... So there are subtle things happening that we like to understand. I also like to point out that LUCA is used in evolutionary biology as an acronym for the last universal common ancestor. So the living thing, the first living thing that gave rise to all other living things. So maybe that's why LUCA is involved in so many projects. So organisms are obviously difficult to understand, so we take baby steps. We try to first understand and answer some of these evolutionary questions in the context of cellular systems. And obviously there's a long history about this genotype-phenotype mapping and understanding it in the context of RNA structure or protein structure. We sort of took the next step of asking very similar questions for cellular systems. And obviously, we already heard that we can abstract cellular systems as networks of interacting elements. So you can imagine there's a bunch of different networks, and some of these networks do different things. So the question is, how does evolution move in this space? Okay, so let me make this a bit more specific. So you can think about cellular networks that mediate some particular cellular behavior, some form of information processing, if you want to call it that way. So here are two examples. One is from chemotaxis, a textbook example, and the other one is from yeast osmoregulation. These are arguably the best studied signaling systems, or if you wish, information processing systems in microbial cells. So very briefly, chemotaxis involves this kind of random bias walk. So the bacteria will swim around, and every now and then they will make these tumbles, which will cause them to change their direction, and then they will run again for a period of time before they change their direction again. So if you put some attractant into the media, you will observe that the bacteria will go towards that attractant through manipulating this process of swimming straight and tumbling and changing direction. So the underlying signaling network for this system is very well studied. And it turns out if you look at the dynamics of that system, I don't know if there's a... Yeah, if you look at the dynamics of this system, you will observe this kind of thing. If there's an attractant in the media and you measure a key protein in that system, this will be the temporal dynamics you get. So pay attention to this part where we have a step change in the signal, and the system responds to that step change, but then goes back to its original level. So that's called an adaptive response, nothing to do with biological adaptation, but adaptive response over time. And a similar response is seen in the yeast osmoregulation system, but this time it involves the volume of the cell. So we were interested in understanding how these systems, networks evolve that provide this kind of adaptive dynamics, which seem to underline this physiological behavior. And people have already been thinking about what this adaptive dynamics might mean, and especially engineering folk have looked at this and concluded that this is actually an implementation of what is known as an integral negative feedback control. Okay, so you have a system that you measure, and you like that system to be at a certain level. When that system deviates from that level, you take that as an error, you integrate that error, and you feed that back into the system as a negative feedback, and that will bring you back to this original state. Now, this is one particular sort of control mechanism you can implement, but it gives you two features which are very interesting. One is that kind of integration, integral negative feedback loop, is very robust. So you can put any parameters on it, but it will always give you perfect adaptation. And it is, those are the two features. It's very robust, and it gives you perfect adaptation. And people then looked if chemotaxis does have those things, and it turns out it does. It does have a negative feedback loop to start with in terms of the biochemistry that implements this. And it provides very perfect adaptation, and that is quite robust if you change the kinetic rates in the system. So I'm just realizing I have too many slides, so I'm just going to keep going fast, so bear with me. So we were interested, this is all fine, but here we have a complex network that implements this adaptive dynamics. So the question is, well, this obviously cannot just come about from the outer space. So how do you get there? So what could be alternative systems that gives this kind of dynamics? So to try to answer that, we set up networks sort of that are evolving in the computer. And in particular, we implemented those networks in cells and let the cells do chemotaxis. So these are results from those types of simulations. You start cells over here. You let them swim around due to the output of their cellular networks. And these are the generations. So over time, you find some cells start to do chemotaxis. So you can then quantify that level of chemotaxis and look what kind of networks you have evolved. And to our surprise, we didn't find adaptive dynamics, but we have found ultrasensitive dynamics. So I will not go into the detail of how this works, but we could then use even simpler models to quantify that this kind of response can indeed lead to chemotaxis. And its performance of this non-adaptive dynamics depends on sensitivity, and so does the performance of this adaptive response. So we had this sort of evolutionary idea that as sensitivity of the system evolves over time, there might have been a time where ultrasensitive dynamics might have been preferred solutions to chemotaxis. So this gave us this idea that maybe ultrasensitivity is important, but we were using, especially in those evolutionary simulations, very simple abstract models of how these interactions in cellular networks work. So we were interested in understanding how these different strategies or responses might be implemented in real biological systems. And ultrasensitivity is actually very well known to be all over the place in biology. So there are different ways of implementing ultrasensitivity. There are these MAPK signaling pathways that we heard of, and sort of feedback loops in transcription, and so on. But in bacteria, some of these systems are present, but if you look at bacterial signaling that underlines something like chemotaxis, it doesn't work like in the MAPK system. It works through these so-called two-component systems. And these are slightly different because they use a different biochemistry. So instead of using ATP-mediated phosphorylation, they use these phosphotransfer reactions. So there's a receptor which gets autophosphorylated upon some signal, the receiving of a signal, and that phosphor gets transferred to another protein that, when phosphorylated, acts as a functional protein and does something, gene regulation or whatever it is. Interestingly, the dynamics you get from these kinds of systems is very simple, and you can't imagine having ultrasensitivity or bistability unless you start to sort of write very complex reaction schemes involving sort of dead-end complexes and things like that. So we wanted to understand what evolution can make with these kinds of two-component systems. And obviously this is, yes. So evolution has to work with what is there, right? That's what the figure is trying to represent. So you have different kinds of architectures when you look at these two component systems. In particular, those kinds of transfer reactions can be implemented between different domains. So you can move domains around and you can have different configurations of phosphotransfers. So you can have multiple domains, for example, then you end up with these kinds of relays, or what people call relays. Or you can have two different endpoints where the phosphor can transfer and so on. So the question was, why do we have these kinds of architectures? What is happening here? What kind of dynamics are possible? I'm shooting for 15 minutes. Is that fine? Okay. So the way we went about this is basically, it is similar to exploring this genotype-phenotype space, right? So I have, obviously I have constrained it largely, but I do especially want to look at these phosphorylase systems where you can have all kinds of phosphotransfer reactions, and all of them can go backwards as well. So these represent different domains where the phosphate can go. And you can now say, well, I can enumerate this system in terms of the parameters. And obviously, I can set some parameters to 0 such that some of the transfers don't happen. So we wanted to understand if we do that, and you can say that these things then will correspond to some topologies. We want to look over topologies and what kind of dynamics that they can do. And particularly, we're interested in this ultrasensitivity because we think it's involved in chemotaxis. Or in general, it's an interesting nonlinear dynamics to look at. So we want to characterize this sort of final step here and look whether it behaves sort of in a saturating manner or in a nonlinear sort of ultrasensitive manner when we input a signal at the top. So, and we can sort of simulate multiple parameters. So we created sort of thousands of systems for each of the topology, and you just classify them. So we were sort of doing on this thing, and then some mathematicians came along and they said, what are you guys doing? We can just solve these systems and tell you what the answer is going to be. And that was great because they could really solve the systems. But because before I give the punch point, what we were seeing from those simulations is only few topologies gave rise to nonlinear dynamics, and only in certain parameter regions. In particular, we were finding that there was this reverse phosphorylation reactions that needed to be high compared to forward phosphorylation reactions. So this black here represents the output. It's sort of linear saturating here, nonlinear over here. And that's exactly what the mathematicians found. They found these conditions on the parameters that were sufficient, that were required, for those response or the output of the system to be sigmoidal. So that was great, and that was confirming that these reverse phosphorylations needed to be larger than the forward phosphorylation. So then we looked at the sort of simplest system that can implement that kind of reverse phosphorylation, a system with two endpoints, which then can reverse phosphorylate. And we could, again, mathematically show the conditions for the nonlinearity here, but also experimentally show it, because this system is small and simple enough we can reconstitute in a test tube. So, okay, I'll have to rush through this, but this is the fun bit. So the other interesting aspect in biology of these systems is that there exists some phosphorylays where each of these domains are encoded on a different protein, and there are others where these domains are encoded in the same protein, so like here. So I can combine the first two steps in one, or the last three steps into one. It turns out that this system does not allow any multistability, but when you combine things in a single protein, then you have multistability possible. So working together with the same mathematicians, we could, well, actually first we could intuitively understand what is going on here. So we took that, the simpler system, the simpler system, and sort of took away reactions one by one until we lost bistability. When you do that, you end up with this system, which is the simplest system. You can still have bistability, and there you can then start getting an intuitive understanding of how that bistability is achieved. So you basically have 4 states of this 2-domain protein, and you can sort of write down the reaction cycles, how this system goes between those 4 states, and you can start to understand where the bistability comes from. Yeah, overestimated my ability to rush through slides. But the point is that what we could then mathematically prove is that as you increase the number of this multi-domain top protein in the system and connect them to the same downstream protein, we can reach unlimited multistability here. And the multistability scales up with the number of systems you can hook up to the same guy at the bottom. And here's the relation with the number of steady states. For n systems, you get 2n plus 1 steady states in this kind of system. What it does so this is very interesting mathematically because you can get the multistability assuming that the signals to all these things are the same. We don't really know what that means for a bacteria, but we can also show that if those systems were receiving different signals, then you can easily implement logic gates in the system. And this is thanks to Luca that he sort of looked at these and provided us with some parameters where we can easily show that these systems can do AND or OR gates and so on, and it also sort of functions like adder, which interestingly, experimentally has been shown, at least in one system, that this system acts as an adder. So I have final few slides here, if I can borrow 1 minute. So where we want to go is really, so we had these evolutionary simulations where we use really abstract systems, and then we had these sort of genotype-phenotype mapping studies where we try to understand what these biochemistries really mean. So ideal situation is to combine the two things, right? And somebody already used this quote, so everything must be simpler as possible, but no simpler. So what are the sort of aspects that we would like to have in the evolutionary simulations? And if we input those aspects, how are we dealing with the evolution? And we've been recently trying to work with rule-based models because we think they better deal with the complexity, and we try to put the rule-based modelings in an evolutionary context, sort of to try to encode these kinds of models in a genome and evolve the genome to get sort of complex structures that better represent biochemistry, but obviously much more difficult to analyze. So with that, I thank you all, and especially Lucas. Happy birthday.